Alrighty, so this is Gram Staining by Jordan Chrisbaum, Michaela Elwood, Katie Groberick, and Jamie Johnson.
So what is a gram stain? Gram staining is a way to distinguish and classify bacterial species into two large groups. These groups are called gram-positive bacteria and gram-negative bacteria. Another name for gram staining is the Gram method. This was named after the Danish bacteriologist Hans Christian Gram, who developed the technique.
What is a gram-positive bacteria? Gram-positive bacteria have a thick peptidoglycan layer and an inner membrane. These bacteria lack an outer lipid membrane, so they are more prone to lysozyme, which is an enzyme that attacks bacterial cell walls. The cell wall or peptidoglycan layer of the bacteria is exposed, making it accessible for lysozyme. Gram-positive bacteria also contain teichoic acid, which provides rigidity to the cell wall by attracting cations such as magnesium and sodium. Gram-positive bacteria appear purple under the microscope.
What is gram-negative bacteria? Gram-negative bacteria have a thin peptidoglycan layer and two outer membranes. It also contains lipopolysaccharides, which protects the cell from certain kinds of chemical attacks, increasing the negative charge of the cell membrane and helps stabilize the membrane structure. Gram-negative bacteria appears red or pink under the microscope.
What are the stages of gram staining? First is crystal violet, second is iodine, third is decolorization, and fourth is saffron. Step 1. Crystal violet. Adding 2 to 3 drops of crystal violet on top of the bacterial smear, let it incubate at room temperature for 60 seconds. Then rinse the slide off with deionized water. The purple of the crystal violet is what gives the gram-positive its purple color.
Step 2 is iodine. Without drying the slide, after rinsing the slide off with deionized water, add 2 to 3 drops of iodine onto the bacterial smear and let it incubate for about 60 seconds at room temperature. Then, again, rinse off the slide with deionized water.
Step 3. Decolorization. Hold the slide at a 45-degree angle away from you and add the decolorizer drop by drop until there is no more crystal violet washing off of the slide. This should take about 10 seconds. Immediately wash off the decolorizer with a lot of deionized water. Never spray the deionized water directly onto the bacterial smear. Instead, spray above the smear and let it trickle down the slide.
Step 4. Saffron. Add 2 to 3 drops of saffron to the bacterial smear and let it incubate at room temperature for 60 to 120 seconds. Then rinse off the slide with deionizing water. Finally, gently blot off the slide with a paper towel and observe the bacteria under the microscope.
Now, what causes the purple color on gram-positive bacteria? The purple color of the crystal violet stains the peptidoglycan layer of gram-positive bacteria. The iodine is what solidifies the color onto the peptidoglycan layer. The purple color does not wash away with the decolorizer because the peptidoglycan layer is so large and it is not easily penetrable by the solvent.
What gives pink/red color of the gram-negative bacteria? Crystal violet does not stain gram-negative bacteria because of the outer lipid membrane. This makes it so that the purple color could not get through the lipid membrane into the peptidoglycan layer. Gram-negative bacteria are able to be decolorized because they have cell walls that have so much thinner layers that allow the removal of crystal violet. The gram-negative bacteria are stained pink/red through the use of a counterstain, such as saffron.
Thank you for watching our video on how to gram stain. I hope this helped.